wherever the sound's coming from. It's very strange.
Yeah, I feel like that's like kind of creepy, but okay. I expected to have a video on me.
Are we waiting for more students?
Yes,
Okay, all right, so I guess eat.
Joseph's supposed to be, yeah, we're relaxed.
Okay.
Joseph's supposed to
I'm
show
gonna get
up.
my water.
Sean can't make it, and y'all got Emma's email,
Mhm.
so she can't make it today.
What are your like backgrounds in, like bioengineering perhaps, but like specifically, just like, this is a very broad topic, so,
I'm bioengineering and applied math.
okay.
I do mathematical modeling, so. General systems biology systems, so biochemical reactions.
Cool.
But I'm focused more on the methods to calibrate them to experimental data.
Okay, cool. And you do cardio,
Yeah, but my background's in math.
okay.
Sorry for my voice, because I'm the stage coach, so my voice is weak.
And then also cardiac stuff, but focused more on like protein-protein interactions and kind of like the muscle biochemistry, I guess.
Okay. So this will be very different.
Yeah, should be
Okay.
great.
I'm okay, since I think I'm the only speaker today.
Yes,
If you want to just like interrupt me and ask questions,
Sounds great.
this is our first in-person
Yeah.
meeting, and so, I wanted to just be very small and very controlled, see how it goes, and get feedback from y'all about what you want going forward. As far as I can tell, I'm only gonna have one more meeting in May, and then June, right before finals week. We are thinking about having a lunch at the faculty club with you and your mentors, so that you can eat together and then talk about your work and your, future, what you want to do and go forward. Who would you invite?
To the
Which
lunch
of
or-
your mentors? You can invite all of them.
Probably Andrew McCulloch.
Okay. Professor McCammon, did he retire?
He's technically retired, but also still very much affiliated with the lab.
Okay.
He's still around, and so I don't talk with him as much, but I could also invite him.
Yeah, you can invite him to see what he says. Nate, how about you?
Probably just had me, my main advisor.
Padmini?
Yeah.
Okay, all right. And Kristen?
Bella's also. My main advisor, Bella's also.
What's she saying?
Her main advisor, Daniela Ellis, also.
Oh. That's all right.
Yeah, no problem.
No, thank you. Marcus is a good interpreter. And since I forgot that Emma couldn't make it, and since your guest didn't show up, we're gonna have lots of leftovers, so y'all make a plate to go. And we'll feed Doug too, Irene.
Oh, yeah, that'd be great.
Make him come up here a third time. I'm sure that's what he wants. I'll email Joseph.
So who's next to come?
Joseph Sawires.
We just give him like 5 minutes and then we start, or you want to wait for him?
Doesn't matter to me.
Well, up to the group.
Give him 2 minutes.
Yeah. People have to.
He might have gotten lost. I don't know. I don't think he
Yeah.
comes over
Did
here.
you guys
very
walk
often.
He has to
over?
come from Chem, so it's
Let's go try.
And you
Yes,
guys,
come from where?
the chemistry building.
Oh,
oh,
okay.
you guys came over, was it, did walk over from over there? I used to work at chemistry. But
That's where Joseph is.
yeah, I know a lot of the chemistry faculty.
Got it.
I used to work there. Who's your chemistry faculty advisor?
I'm not in Chem.
Oh, you're
I'm in-
not, Joseph.
Joseph is the
Yeah,
one.
yeah, yeah. He's
Yeah,
at,
speaking of which, there he is.
Okay,
He's just outside. He said.
Oh, he's not there. Oh,
he's parking his bicycle. Did you get the one with the chimichurri?
I work with
Okay. Well, you get whatever you want, but I figured You'd like the chimichurri. Hey, Joseph, the food's right there. Water's right there. This is Liana, our guest speaker. That's Joseph Sawwires.
Oh, yeah.
Okay, now you know. Emma's not coming today and Sean's not coming today, so. Sean has, was it his defense? What did he say? Maybe advance, yeah.
You can bring it in, I'm sure.
What's your mic?
I think it'll also be fine out there, but.
Or put it in the patio, right there. Is it open?
Yeah, candidacy exam. Scheduled today and tomorrow.
Let's bring it in here. You feel more comfortable with that. We see someone on the cycle.
That's true, I'll watch it.
Well, we'll like. We'll.
I ain't doing nothing, cause I'm too slow. I would be no help.
So we can't volunteer people running that?
No.
Okay.
Is there a what?
Other plates.
Yeah, on the right-hand side, there's not any plates. Did somebody grab more than
Yeah,
one?
there's more here. Sorry, what am I called? Here you go. I didn't touch it.
Wait,
There's
are
two of
you
them
in
here.
Rob
Please,
Mike's
if you want
lab?
us-
No, but I collaborate with Rob a lot.
Okay,
Yeah.
gotcha. Like, I feel like that's not a crazy assumption to make based on something that
No,
way, but
I mean, like what he does is metagenomics,
Okay.
and so I will actually talk a lot about that, but I'm more interested in like proteomics.
okay, yeah, I guess I could have read a little more.
Yeah,
closely.
no, I mean, literally Rob will probably be on every single thing I do. But yeah.
No, okay, that makes sense. That's cool.
Joseph, we've got plenty today, so, you know, don't feel bad about getting up and getting a second helping, any of you, so. And then you can make a plate to go.
Well, I told my wife I would if there's extra.
There you go.
She loves Mendocino.
It's-
What lab are you in?
I'm in David Gonzalez's lab and Rich Denneman.
Why have I heard of David Gonzalez?
He is like the he does multiplex proteomics, which might be why.
I don't know anyone in the bio department. Does anyone in there? This is David Gonzalez.
Not that I know of.
No, we had a previous person who's like graduated like 2 years ago. Should I just start?
Sure.
Okay, so I said this previously, but since you just arrived, interrupt me. I like prefer that, ask questions as we go. I wasn't actually sure like the background of the people that I was going to be talking to today. So it's a little bit more general. I'm also second year, and so I have a lot less presentable data at this point. But yeah, I'm happy to answer like any specific questions, anything that you're interested in. It's a really broad topic. So today I'm going to be talking about stool metaproteomics to uncover gut microbial protein effectors in disease, kind of the past, a little bit about the present, what I'm working on, and what I hope to work on for the rest of my PhD work. But I'm also in the biomedical sciences program. So I want to start with this quote credited to Hippocrates: "All diseases begin in the gut." I'm classically trained as a neuroscientist, but got to UCSD and got really fascinated in the gut. And I think that a lot of neuro diseases really start in the gut. So that's where I'm moving to. So this is an outline for my talk today. I'm going to give you an introduction into stool metaproteomics, since it's probably a brand new term for you. I'm going to talk about the past work from the Gonzalez lab, how we use stool metaproteomics to identify druggable targets in ulcerative colitis. I'm going to talk a little bit about the present work that I'm doing, expanding the meta in metaproteomics and addressing some of the limitations in the field, and then how I plan to bring all of this together and apply it to study the gut-brain axis, specifically Parkinson's disease. And then I'll summarize my work. But I put questions at the end in this outline. Again, interrupt me. So what is stool metaproteomics? It's an emerging mass spectrometry technique that we can use to study the gut microbiome. So there's a lot of proteins in stool, a lot of human proteins, and even up to 50%, kind of depending on the sample, of stool can be microbial content, microbial proteins. And so we can basically prepare those, put them on the mass spec, sequence them, and get a lot of interesting biology from that. Maybe you're asking yourself this question, why stool? I ask myself this almost every day as I'm preparing.
As we eat lunch.
Yeah.
Sorry about that, y'all.
I know, this is like, I really had to develop a strong tolerance. It's not very glamorous work, but it is important. So first off, stool, there's no pictures of stool in this talk also. It's biologically relevant and correlative. So looking at stool can give you really important information about the gut, which is of course important when you're trying to study health and disease states. There's a surplus of sample from every collection for processing, which allows you to be able to do a bunch of different things with only one collection, which is really valuable when you're studying humans because it can cost a lot of money just to get patients in the clinic once. It's completely non-invasive. Even a blood draw is more invasive, so it's really the least invasive kind of biospecimen we can take from participants. You can also do really easy repeated sampling, for example, like longitudinally as someone's aging or before and after treatment. And it's the most complex biospecimen that you can take from someone. There's so much content in stool. A lot of it we really haven't even explored as a field. And that's something that I want to develop more during my thesis work.
I had a quick question.
Yeah.
So, like out of all those things, I guess the last thing you're saying is most complex, like how that, I guess, compared to like tissue, because I guess tissue is usually also a sample that we usually take. You would say like stool is more complex?
Yes, because so you can have up to 500 or more bacterial species alone in stool, all the human proteins, and then you get metabolites, proteins, DNA, transcripts, food proteins. So it's complex in the way that there is just so many more things that need to be parsed through, and a lot of it is left unannotated. So tissue is also like complex in the way that there's a lot of things that you can ask with it. But I think complex in terms of stool a lot of times means complicated. So just an example of the types of host proteins, so human proteins that we can detect in stool. Surprisingly, a lot of it actually isn't even like digestive tract proteins, although a lot of that content is there. So like colon, small intestine, spleen. But we can also get things like blood and liver and even things like lung proteins. So stool can be used to monitor not only like gut diseases, but also diseases like of the liver, general immune diseases, and as I will eventually do to study neurodegenerative diseases. So we talked a little bit about metagenomics. And metagenomics and 16 RNA sequencing are techniques to basically look at microbial genes and then from there quantify their composition in a complex system like stool. But looking at genes doesn't necessarily correlate to the proteins that are there. Proteins are our main actors within our body. And so there's a lot of additional pros to looking at the protein level or metaproteomics versus looking at the genome level or metagenomics. For example, you can get better quantification information from looking at proteins. From genes, you can predict the protein content that's there, but you can't always quantify it. It doesn't always correlate. So it's better or more accurate to look at the protein level. You can also get more information about function, signaling pathways, which is really important when you want to understand basic biology of a disease or of a healthy state. You can get a lot more information about post-translational modifications. Most of this is not encoded necessarily in the genome, but can be really important in terms of like functional consequences for a system. And you can get information on protein-protein interactions by looking at the protein level. And lastly, and more translationally, you can get druggable targets. Proteins are super druggable, much more druggable than looking at a specific microbe of interest or a set of microbes. And also biomarkers to look at onset of disease or track progression of disease or remission. So this can be really valuable kind of at a basic biology standpoint, but also at a translational standpoint. So now I'll show you kind of how we in the Gonzalez lab have used stool metaproteomics to study ulcerative colitis and develop kind of a preclinical therapy that is now being more sophisticatedly studied by another graduate student in our lab. So we performed multi-omics on ulcerative colitis, which is a type of inflammatory bowel disease. And this included stool metaproteomics, but we also did serum proteomics, metagenomics, metabolomics, peptidomics to really get a full picture of what was going on. But where most of the information came from was with our metaproteomics. This is where we kind of found the most interesting phenotypes. And so if you look here, so this is a quantification of the number of proteins that correlate with disease severity in 2 different cohorts of ulcerative colitis. And you can see that there's a lot of Bacteroides proteins that are strongly correlating in both cohorts with disease. And so we looked more into what these proteins actually are, and they're primarily proteases, Bacteroides proteases. So then we went to mice to test this hypothesis that proteases might be driving ulcerative colitis disease severity. We colonized IL-10 knockout mice, which is a colitis model with Bacteroides fulgaris, one of the primary Bacteroides that popped out for us. We treated them with and without a protease inhibitor and then monitored their inflammation, and we found that the protease inhibitor strongly reduced inflammation. And so this was our very first kind of proof of concept, going from metaproteomics to a druggable target to a drug candidate that we're now kind of fine tuning in the lab. Yeah.
So what are the cohorts and how many patients?
Yeah, so I did gloss over that. So the first cohort we did was just ulcerative colitis only, and then eventually we scaled up to over 200 patients, with half of them being ulcerative colitis, half being Crohn's, which is the other type of inflammatory bowel disease, and then a small cohort of healthy individuals.
Okay, and then could you re-explain how you got to a druggable target?
Yeah.
With the, like, which protease inhibitor, or is it just any protease inhibitor?
It's actually like the protease inhibitor, if you've ever done a Western blot, like the Roche tablets, which I don't really know how they got that to be approved on our animal protocol, but it is. And yeah, so it's just a general proprietary protease inhibitor, which is why now people are looking at to see if it's like serine proteases and other kind of parsing that phenotype out. And I think you bring up with your question a really good point is sometimes with this type of data, you just get so much to follow up on. And I think the grad student who's really leading this saw this, this really big change in Bacteroides proteins in general, and then start to follow up on that. So there's probably a lot of other stuff that you could pull from this data, but that's really how he narrowed down what he wanted to follow up on.
And then also, is IL-10 just an inflammation in general, or is it specific to gut diseases?
There's not really any great colitis model in mice, and IL-10 knockouts induces inflammation in the colon. So it's like a whole systemic knockout, and that is one of the phenotypes that happens, but yeah, it's not a perfect model. And there's a lot more data to show this rescue that I'm not showing. And the other thing that I take away from this study is that metaproteomics is incredibly powerful. So looking at all the other kind of omics that we did, metaproteomics is the best at predicting disease activity. And you can see that here, looking at the mean squared error of disease prediction, and it performs as well as all the other metrics individually and as well as all the combined data. It is also really correlative with all the other data that we looked at. So metaproteomics is sufficient to predict disease activity, and it correlates strongly to all the other omics that you could look at when you're interested in these types of questions. So why not always do metaproteomics? There's
One
a
quick question.
yeah.
I should do. The MB is
Metabolomics.
Okay.
Sorry, yeah.
Perfect. And then did you try to use metaproteomics in addition to the other combined data to see if you can do even better? Or do you know if the study did that?
Like a combinatorial.
Because you have like
type
the
thing.
combined data, right, which you're saying it's doing just as well as, so if you
Yeah.
did the metaproteomics with the combined data, you get even better correlation potentially.
Oh, the combined data also includes
Okay, I wasn't
metaproteomics.
sure about that.
Yeah,
Gotcha.
so basically none of the other data increases the prediction
Yeah, that makes
value.
sense. Okay.
Yeah.
Cool.
So there's a lot of limitations that are left in the field of metaproteomics. It's a very new field, especially stool metaproteomics. A lot of people have been using metaproteomics to study like soil samples, for example. So one thing that I've been working on in the last year in the lab is kind of expanding metaproteomics, specifically to include more than just the bacteriome, which I showed you a study about. So some of the current limitations, it's low throughput, and there's inadequate contaminant removal. These are big issues when you're working with stool. You don't want to be working with stool for years and years. And if you don't properly remove the contaminants, it leads to instrumentation downtime. Subjects are very variable, and this is a huge bioinformatic hurdle to overcome, because if your healthy controls are too different, then you'll never be able to pull out things that are related to disease. There's also limited kind of reference databases and limited resources for understanding post-translational modifications. So today I'm going to talk about these three. If you have interest in the other two, I'm happy to talk about them, but for time I removed them. So the first way that we are addressing throughput and contamination removal is with this stool high throughput proteomics pipeline, which a postdoc in our lab actually named Ship Pro. And you have to have a good sense of humor if you're going to be working with this type of sample. And it is published with this name, so it is out in the world now. The workflow kind of very quickly works like this. So in a 96-well format, we feed beat our stool samples to basically lyse up the cells, and we use SDS, which is like a really harsh reagent, which helps kind of make the samples extra accessible, make the protein extra accessible to us. Then we load them onto a special type of column to isolate the proteins, and then we do a digestion with trypsin, which cleaves the proteins into peptides in a very predictable way. Then we clean them up on a C18 column to additionally remove any contaminants. And then special to the Gonzalez lab is this multiplexing workflow, where basically we add a mass tag to a bunch of samples. We can mix up to 16 samples at the same time and then run them all on the mass spec at the same time. And this helps us identify more proteins across all the samples and get better relative quantification information. We also will fractionate our samples, going from like 1 multiplex to 24 different samples, which makes the samples overall less complex when they go onto the mass spec, giving us more ability to identify what's there. And then we perform our tandem mass spectrometry, which gives us precursor information, peptide identification information, and also quantification information at the MS3 level. And then the last part is kind of where we have the most work to do, which is in searching the databases and then identifying what is there. So again, like I said, stool is incredibly complex. And so there's a lot of unannotated spectra where we're kind of losing potential information, especially when working with such a precious sample, like a human sample. It's really important to be able to get as much information out of it as possible. So like I mentioned previously, what people currently look at is human and bacterial proteins, but I am working to expand different reference databases and validate them so that people can have that available as a tool, so you can answer new and exciting questions. So I'm looking at food proteins. There was this really interesting study that showed that anytime you search, the C. elegans proteome, you should also search the E. coli proteome because they eat E. coli. So if we do that for C. elegans, why aren't we doing that for humans? Now, obviously, we eat a lot more complex things than just E. coli. So this has been a particularly challenging problem, but the field of metabolomics is actively addressing it. And so proteomics, hopefully, will be not far behind. We also have fungi in our guts. We have a mycobiome. things like different yeasts. And so it's important to include those as well, even though they're not nearly as abundant as bacteria or archaea. And lastly, we have our own virome. And the virome is primarily bacteriophages, like 98%. And that can vary, if we have an active infection. But these are still adding proteins to our stool and would be really important to be able to annotate and understand the biology of that in the gut. So some of the things that I found with my new reference databases, I took 10 different species of microbiota based on their abundances in the Human Gut Microbiome Project. And then I searched some spectra that I had from an ulcerative colitis study that I performed separately from the previous one I showed. And I was able to identify over 250 proteins from these different species. One that I particularly think is fun is this Saccharomyces cerevisiae, and this is beer yeast or brewer's yeast. And then I compared the protein abundances that I found in ulcerative colitis to the abundances I found in my control samples and found that almost 60 proteins were actually down in abundance in ulcerative colitis, which can be really important to understanding the biology, and has really not been looked at further in the field. No one has looked at microbiota proteins in ulcerative colitis, and people have just briefly looked at microbiota composition using genomic levels. I'm also looking at food. This is really hard to parse out because a lot of foods are very similar. And we have a really similar proteome to pigs, beef, goats, things like that. So this is kind of a computational obstacle I'm working on right now, but some preliminary data I've been able to find almost 2,000 food proteins, start to identify pig proteins, beef or cow dairy. You can't identify actually the source of a cow protein if you find it in your stool, whether it came from the meat or from dairy. And most of the individual species contributions are from animals. And then when we look down at like different groups, we see red meat and white meat kind of come out first, but then also some other food groups like grains and grass, legumes, fleshy fruit, and vegetables, fungi, which also include beer yeast and baker's yeast, and then some other food groups as well. So this is something that I'm still working on, and hopefully will be going out within the next few months. And lastly, very quickly, what I'm going to be doing. So like I mentioned, I'm a neuroscientist by training. So everything that I've been developing is really to understand the gut-brain axis. And so my disease of interest is Parkinson's disease. So Parkinson's disease is primarily a motor disorder. It's a neurodegenerative disorder where you lose dopaminergic neurons in the substantia nigra. And so if we look at this brain here in the midbrain, we'll slice it, and then that is what these images are. You can see the substantia nigra are these nice melanated neurons, and then when they die in Parkinson's disease, you completely lose this, this cell population as indicated by the lack of neuromelanin. One of the primary hallmarks of Parkinson's disease is alpha-synuclein forming protein aggregates. So if we look at a nice healthy substantia nigra, we see all the cell bodies, the neuronal cell bodies, and then we lose these cells, they die in Parkinson's. Then if we zoom into the one cell that's kind of still hanging out, this right here is a Lewy body. So that whole thing is a massive protein aggregate and is one of the reasons why there's cell death in Parkinson's. So Parkinson's is really classically thought of as a motor disorder. And once you start to develop motor symptoms, that's when you can receive a diagnosis of Parkinson's. However, up to 20, 30 years before any motor symptom onset, you can develop gut dysfunction, including gastroparesis or constipation. And so this is kind of our first clue that gut symptomology might be underlying Parkinson's disease onset and progression. And so people have been really interested in this in the past 5 years or so and have done a ton of sequencing of the Parkinson's microbiome. And a recent meta-analysis shows some consistent changes in different taxa. So things like lactobacillus, that's a really common probiotic, and bifidobacterium, also a really common probiotic, seem to be consistently up in Parkinson's. And then a whole set seem to be consistently down across over 20 studies that this meta-analysis looked at. But of course, none of these studies really show us, well, should we be taking probiotics? Should we have certain antibiotics target certain bacteria? We don't really know. So I'm really interested in looking at specific proteins. Are there microbial pathways that are going up or down that we could kind of more specifically target versus targeting a single taxa? Because like I said, lactobacillus and bifidobacterium are common probiotics. They're thought to be good for us. They're some of the first bacteria that colonize our gut during development. And so they also seem to be up in Parkinson's, and that doesn't necessarily reconcile. So I have this massive test cohort of 157 participants, 65 with Parkinson's disease, 65 aged and sex-matched population controls, and then this third group of about 27 spousal controls, which is a really exciting group since 90% of Parkinson's has no clear genetic cause, so it's likely to be from behavior and environmental exposures, which oftentimes household controls will share. So hopefully the next time I talk to you, I'll have some data from this. I'm actively processing these samples. So as you can imagine, that is so glamorous. Yeah, and then with that, the main takeaway points that I want you to have from my talk is that stool metaproteomics can be used to study basic biology, number one, and then also can be used to identify druggable targets and biomarkers for human disease. And this can even be applied to diseases that are not classically gut. People have also done liver studies, and I'm hoping to expand it into diseases of the gut-brain axis. And then second, despite its utility, stool metaproteomics has a lot of limitations the field needs to address, especially as more and more people start to move into the field. I'm hoping to address some of these. Other people in the field are doing other important work. But even with all of that, I think there's still a lot of promise, and it's already proved to be really useful. And with that, these are my labs. This is me processing some stool. It's always really fun. Yeah, and I'd like to thank my labs and my funding and support. Does anyone have any other questions?
Oh yeah, I had more of a general question. Like,
Mm-hmm.
could you explain more how you go to a specific druggable target from like the 2000 proteins that you're looking at, like you look at food proteins and
Yeah.
proteins?
I mean, that's so hard in actuality. And I think basically you can do some statistics to see, which proteins seem to be more correlated with different, disease phenotypes or with disease in general versus lack of disease. That helps narrow it down. But still you can be left with, if you're looking at the whole microbial proteome, you could be left with, 30,000 proteins in total, that then you narrow down to 2,000 proteins. So still a lot. I think part of it is brute force, looking at the literature, seeing what seems, maybe the most interesting, whatever stands out the most statistically, things that are the most feasible. Some of these microbes are really, really hard to culture, or there's not a lot known about them. So those you might not want to follow up on. But yeah, there's really no single path. It's kind of whatever you're interested in, whatever seems the strongest, whatever also seems the most feasible. So that will be an obstacle that I have to overcome in about 6 months when I start to follow up on Parkinson's things. Yeah.
This is more of just kind of like a general proteomics with the gut, but in terms of kind of your quantification of the amount of protein or whatever, are there specific baseline proteins that you guys look at? Obviously, I know that isotope labeling is really big for mass spec in terms of looking at relative changes, but you're not probably going to do that at least with human studies. Obviously, I guess you could do that with animal studies, but I'm just kind of curious how that workflow goes and how do you kind of really identify those significant changes, because there is probably so much variability in the gut.
Yeah, that's one of the things that doing multiplex proteomics can help to overcome, where anything that's run on the mass spec at the same time, we can relatively quantify to one another. And so from this specific workflow, you're not able to say like the absolute amount, like you could with a Western blot, which is how we would validate this. You can say that overall, this group is higher than the other, which can still be really useful.
Yeah.
And in terms of like, biomarker development, then you would want to look at like a kind of ratio so that like it would actually translate into the clinic, of something going up or something going down. But yeah, you could use some kind of benchmark, but Stool is so variable that I don't even know if that would be possible.
Sure.
Yeah.
No, that makes sense.
Thank you so much.
Thank
Appreciate you
you.
being
Thank you all
willing
for
to
listening.
Actually, I have one other question that's more far out, but do you think that like ultimately using a combination of metaproteomics and metagenomics will be kind of the best way to kind of approach these
Yeah.
microbiome and gut approaches, just because you can get maybe more data that way, but then you can really kind of pick up what's being expressed and what mechanisms are at play with the proteomics side of things.
Yeah, I think, okay, so one thing I didn't talk about is that often metagenomics is integrated into the metaproteomics workflow, that you actually use metagenomics to generate that search
Okay, that
database.
makes sense.
And so, yes, however, metagenomics and metaproteomics, you either have access to one or the other, and they're both expensive. And so a lot of proteomics labs don't necessarily have access to metagenomics.
I
So it's
see.
not necessary to do both. I think it's really useful. And luckily at UCSD, I don't have to choose personally.
Nice.
So yeah, but I think keeping them integrated is the way to go.
I was gonna ask you, so again, what year is this for you?
My second, I'm in the
This is your
my
second
yeah.
year? Okay, and your department is?
I'm in the pharmacology department, but
You're in
biomedical sciences.
Oh, biomedical, yeah, because they draw from different.
Mhm.
Okay.
Yeah.
All right, and then, so then you will have, when will you present your Senate exam? When would that be scheduled for you? Like in 2 more years or something like
Like my advanced candidacy
Yeah.
exam.
that? Like 2 more years?
I have no idea. They haven't told us anything.
Oh,
But probably
okay.
at least a year. So we've had like our minor proposition,
Uh-huh,
and then we have like a major proposition, which
Okay.
is like the advancement. Yeah,
all right. Okay.
probably a year.
Hey, that's great.
Thanks.
Thank y'all for coming. Thank you so much, Liana. I appreciate you being willing to speak with us.
Of
and
course,
present.
it was fun.
Well,
honestly.
good.
And I have a lab meeting tomorrow, so this will just
Oh,
prep
this will
me
be
for
your
that. Yeah.
There you go.
Yeah, I mostly talked about the Gonzalez lab also, but Rich Ganneman is like a blood-brain barrier person. So my other goal is to then track gut microbial proteins from the gut to the brain. So I might have to tap some engineering people to help me do that. So yeah. Yay, I'm gonna leave now.
Do we want to stop recording now?
It's up to you. Yeah, I can stop it now.
Let's go ahead and stop recording. And I just want to talk to